Hi everyone. Today, Alyssa, Beth, and myself, Kristen, are going to talk about the use of recombinant protein in medicine. As you can see, we've got a lot to cover today. We're going to discuss things like what is the purpose of recombinant protein technology, what is it used for, a little bit of history and development, the current applications, advantages and disadvantages, and potential future applications or any extensions. Now, before we get too ahead of ourselves, we need to get an understanding of the basics before we move into the nuts and bolts. So let's talk about proteins. They're the most abundant and versatile macromolecules in the living system and have critical functions in essentially all biological processes. They function as catalysts. They transport and store molecules such as oxygen. They provide mechanical support and immune protection, generate movement, transmit nerve impulses, and control growth and development. Proteins are linear polymers built from monomer units called amino acids. These amino acids are the building blocks of protein. The sequential order, number, and chemical identity of the amino acids determine the structure and function of the protein. Now that we have a better understanding of proteins, let's look at recombination. Recombination is an arrangement of genetic material. It involves an enzymatic process, which is the linear arrangement of nucleic acid sequences in the chromosome are changed by cleavage and rejoining. A recombinant protein is a type of modified protein which has been encoded by a recombinant DNA, which are two segments of DNA which are joined together in a plasmid, generally found in a bacteria. The recombinant DNA is inserted into the bacterial plasmid. These DNAs are translated into proteins and now have new characteristics. Recombinant proteins can be produced in large quantities by genetic engineering. Now that we have an understanding of proteins and recombination, what is its purpose and what is it used for? Recombinant protein technology is one of the most amazing, revolutionary discoveries in the field of medicine. Proteins are often derived from other organisms because it's nearly impossible to obtain sustainable yields from natural sources. This is the purpose of protein recombinant technology. This technology has been widely used in the fields of biotechnology, medicine, food production, agriculture, bioengineering, and in research. It has saved and changed the lives of so many people. Today, recombinant protein technology can be found almost anywhere, from your pharmacies and your medical and testing laboratories, to farms, supermarkets, even in your medicine cabinet at home, and even in the pet store. Next, we'll take a look at some history and development. On this slide, you'll see a timeline, and we go back to the late 60s to where it all started. Paul Berg and Peter Loban independently created an approach to create rDNAs in vitro and using them to manipulate genes across different species. Then in 1971, Douglas Berg and his colleagues isolated the first plasmid bacterial cloning vector. In 1972, David Jackson and his colleagues, Peter Loban, developed the procedure for joining DNAs in vitro. In 1973, John Marrow and his colleagues clone and propagate rDNA genes in E. coli. In 1976, the NIH issued guidelines for the use of recombinant protein technology. In 1978, insulin was produced from the E. coli bacteria This production of synthetic insulin was groundbreaking technology and has really paved the way for the technology to be used in other medical treatments. In 1980, a patent was awarded. In 1986 to 1991, many oncology and antiviral treatments were produced using recombinant protein technology. In 2001, the sequence of the human genome was published, making it possible for researchers to begin developing genetically based treatments for disease. In 2003, glowfish were introduced to the US. With further research, more discoveries are being made so that it's possible to treat and even cure many diseases with this technology.
Hi everyone, Beth here. I will be taking you through the process of protein recombinant biotechnology. So there are three important sites a bacteria plasmid must have in order to successfully transport the target gene. So the first one is origin of replication. So this is the initiation site, and this is the site that actually allows for the cloning of foreign DNA to occur, which is down the bottom of our diagram. On the right we have the multiple cloning site. So this is the area where many target genes can be cleaved here using restriction enzymes that are coded specifically for the target gene we are focusing on. However, for this presentation we are just cloning the one gene, which is insulin. On your left, the antibody resistant region. So this region permits only the plasmids that have the antibiotic resistant gene to grow, as they are resistant to the antibiotic that is present. So there are other plasmids that perhaps have not taken up this gene, therefore they won't actually be allowed to grow inside the plasmid. This is essential for the replication of the target gene in question, and it keeps all the other unwanted material out. So for example, with insulin, the plasmids that have the insulin and also have the antibiotic resistant gene, this is good for us. It means that it will be able to replicate and we'll have many of our target protein, which is obviously the focus. Okay, so steps in protein recombination. And I'm going to use the example of insulin, as it's quite a common protein that I'm sure everyone has heard of before. So the first step in protein recombination is the insulin gene is our target gene. Okay, so in the very first picture you can see there's a little red line, and in between the two red lines is our target gene of insulin. So from here, a restriction enzyme known as endonuclease is used to cleave this DNA of insulin. From here, it is isolated and joined by another enzyme called ligase to this bacteria plasmid or vector. So to make it clear, it's come from a eukaryote cell because it is a eukaryote piece of DNA, and it is transferred into the plasmid, which is a prokaryote cell. So after this process, the plasmid is labelled as recombinant DNA, or also known as chimeric plasmid, which is basically just a eukaryote and prokaryote DNA combined. So insulin is from eukaryote and it is transferred into the bacteria cell called a prokaryote. So here we're going to talk about the transformation from plasmid to host cell, which the most common bacterial plasmid is E. coli. So transformation of the plasmid into the host cell is done through a process involving a heat shock and calcium chloride type treatment. This process requires a starting temperature of 0 degrees Celsius, which then rapidly increases to between 37 and 43 degrees Celsius. And this is crucial for the cells to be able to uptake the plasmid DNA. It's not completely known as to why this process works, but it's definitely something that's being used, obviously, because it is helping with the uptake of the DNA. This is the site where protein synthesis occurs by using the host cell's machinery for transcription and translation of the insulin gene. This results in the production of several proteins derived from the target gene. In this case, multiple insulin proteins are being produced. So in the field of medicine, protein recombination of insulin has been used for patients with type 1 diabetes. So in type 1 diabetes, the pancreas actually doesn't produce enough insulin or cannot produce it on its own. So by using this biotechnology, scientists are able to provide type 1 diabetics with basically a synthetic insulin, which obviously can help them balance their diabetes. So now we're going to look at the applications of this biotechnology. So recombinant proteins have recently revolutionized the field of medicine. They are now a key component of treating life-threatening diseases, and below are a few examples where this technology is being applied. So first one is hematopoietic growth factor. So this contributes to growing blood cells. Tissue and bone growth factors, interferons, so signal proteins which are activated by host cells when pathogens are present. Recombinant protein vaccines, so creating proteins that essentially protect against specific pathogens. And hormone therapy, so synthetic production of a specific hormone for people who lack sufficient natural growth of that particular hormone. So in our case, it would be insulin. So this is especially important for our type 1 diabetics, where they can get insulin provided to them to help them maintain their hormonal balances. So here are some common diseases protein recombinant biotechnology is currently being used to treat. So we have autoimmune diseases such as multiple sclerosis, blood deficiency, so hemophilia or anemia, which is probably the one most well known, so that's low blood cell production. Cancers, so T-cell lymphoma and also bone marrow transplant. Cardiovascular disease, so that's myocardial infarction. Genetic diseases, so cystic fibrosis and diabetes. Inflammatory diseases such as allergies and septic shock. Nervous system disorders, so amyotrophic lateral sclerosis. This is a rare disorder, however quite detrimental as it does impair voluntary movement. And last but not least, we have tissue damage, so the healing of wounds. Awesome, thanks for listening.
My name is Alyssa Mickle, and I'll be talking to you about the different advantages and disadvantages of recombinant protein technology in medicine. I'll first begin with the advantages. There have been many advances made in the medical industry over the years. DNA used to be regarded as one of the hardest molecules to analyse. Due to the development of genetic engineering and the recombinant protein technology has opened up a whole new area of DNA manipulation with outstanding precision. With improvements and discoveries in treatment of disease, identification and tracking of viruses, and drug development and other advances in medicine. To begin with, I'll talk about how recombinant technology has helped us with treatment of diseases. Recombinant protein and DNA studies have led to measurable advances in medical treatments towards what used to be known as incurable diseases. This is with the use of gene therapy. Gene therapy is a technique that takes a mutated gene that causes disease, modifies it, and then replaces it with a healthy gene. This technique has already been used in the treatment of diseases such as cancer, cardiovascular disease, and other genetic disorders. It has made great progress in the treatment of cancers in recent years and is still developing further with more efficient and effective treatments. Gene therapy is also opening up new doors in the treatment of cardiovascular disease by providing myocardial protection, regeneration and repair, preventing the bypass of graft failures, and risk factor management. Not only is it being used in the treatment of disease, but it's also being used in identification of genetic disorders and diseases. Recombinant DNA technology has improved the screening for genetic diseases by analysing DNA directly. By taking a gene and cloning it, scientists can compare the gene of interest to a cloned gene that is normal and identify any mutations that might occur. This technology is being utilised in the detection of genetic diseases in utero. This process is called amniocentesis. Amniocentesis is a test performed during pregnancy to assess if there's any abnormal genes present. The development of amniocentesis has allowed for early detection of mutated genes, which result in a vast range of diseases and disorders, such as Down syndrome. This development has given many expecting parents who may already be aware that they are carriers of certain recessive genes or have pre-existing conditions to test and find out if they will pass it on to their child. It is also being used to track cancer cells. Cancer cells have the ability to hide from the immune system and reduce the T cell survival rate. This allows the cancer cells to spread more rapidly. However, using recombination and modifying the T cells with cancer-specific antigens, it is shown to increase the recognition rate, preventing the rapid spread of tumours throughout the body. Recombinant protein technology has also allowed for many drug developments. DNA vaccines is the new approach to immunization against viruses. Compared to the regular vaccines that are already on the market, recombinant vaccines are lower cost, more effective, and painless to administer. Administered through nasal inhalation, it transfers the adenovirus vectors that are encoded with pathogen antigens through the nasal cavities and into the airways, allowing them to act as anti-influenza drugs. Other advantages in the recombinant protein technology is the development of supplementary hormones. Lispro, or Humalog, is one example of this. It is used in the treatment of people with type 1 or type 2 diabetes, as it's a fast-acting insulin. Another similar product is Epotin alpha, which is used in the treatment for severe anemia and has also been found to cure some cases. It mimics the effects of the naturally occurring erythropoietin, stimulating more red blood cell production in the bone marrow. While recombinant protein technology has allowed for many advances in the medical field, it has also faced some problems and shortcomings of its own. I will now look at the disadvantages of the recombinant protein technology, such as the misuse of the developed drugs and overfolding of proteins. The misuse of the developed drugs is becoming more popular in the sporting world. Many of the drugs developed, especially for gene therapy treatments, are being misused through what's called gene doping. It is an increasing problem in the sporting world. Instead of using recombinant DNA to restore damaged genes or replace the mutated ones, it is instead used to enhance athletic ability. The same recombinant protein, Epotin alpha, which was mentioned just earlier for its use in curing anemia, is used by athletes to increase their red blood cell count, allowing them to carry more oxygen for better endurance. This is commonly known as blood doping, made famous by Lance Armstrong. Another hormone produced through recombinant protein technology that is used in gene therapy is insulin-like growth factor 1. This was developed to treat muscle degenerative diseases. In the sporting world, however, it is being used to genetically dope the athletes. The recombinant protein manipulates the muscle-building gene, enhancing muscle growth and performance. An agency was formed in 1999 called the World Anti-Doping Agency, or WADA. This agency was formed to work against doping in professional sports. One of the ways it does this is by monitoring the developments of genetic studies and analysing how these new drugs can be used for genetic doping. Another shortcoming that protein recombinant technology has faced is the misfolding and aggregation of the proteins. For successful production of recombinant proteins, the correct formation of disulfide bonds is essential. The incorrect formation of a disulfide bond can lead to a protein misfolding and aggregation, results in several reactions, causing the disulfide bond to form in the cytoplasm. If the disulfide bond forms in the cytoplasm, this can cause an inactivation of the proteins. Along with this, aggregation can also lead to an immune response, resulting in the body not accepting the modified gene. This will lead to future problems in developing new recombinant proteins.